So let's see, today is the last instructional day before the exam. On Thursday, we'll have a review, question and answer session like we usually do before an exam, and then next week are the exams. I posted on Moodle options for taking the exams. Things are complicated because of my own schedule with the practical and also with other deadlines such as eCanal. So there are options available to you on Moodle for both the lab practical and for the for exam 3. Please choose the best set of options for you. If none of those options work for you, contact me and I'll see what I can do, but I'm hoping to get everybody into those options. If you're in the Thursday evening section of lab, remember that I'm expecting you to take the lab practical Thursday evening. If that's not a problem, if that's a problem for you, please let me know. So remember to bring your questions and, questions to class on Thursday, leading up to exam 3. We'll talk next week about the final exam. Well, yeah, we'll talk next week about the final exam. The final exam is scheduled for Monday, which happens very soon after your scheduled exam 3, which means I'll be flexible, those of you who want to push it back a few days. But that's up to you, that's flexible. I will offer it on schedule on Monday morning, but I'll also be flexible about when you take it during exam season. Okay, I think that's all the housekeeping we need to do. Let's talk about the female reproductive, anatomy, physiology, and endocrinology. So let's get oriented here. Anterior, posterior. This is supposed to be the spinal cord, inside the vertebra, in the back. Here's this is a mid-sagittal section. Here is the rectum, anus, that's not part of the reproductive system. Let's start up here. Here's the ovary, so we're now in the abdominal cavity, lower abdominal cavity, really inside the pelvis. The pelvis isn't shown. The ovary is the female reproductive organ, the female gonad. That's where the ova are going to develop. Here is the fallopian tube, which is going to bring the mature ova in from the ovary to the uterus. There are structures here, ligaments and special structures here at the beginning of the fallopian tube, which I'll talk about in a slide in a few seconds. So in particular, there's a ligament between the fallopian tube and the uterus that looks like it physically allows the ova to move that way, but that's just a ligament. It's just a connective tissue. The ova moves from the ovary into this wrinkled, in this image, this wrinkled edge of the fallopian tube has this name. It's called the fimbria, and that's where the ova is going to be picked up by the fallopian tube and then transported by sort of ciliary movement of the cells that line the fallopian tube from the ovary to the uterus. The uterus has layers. It is a muscular organ. It's full of smooth muscle. But there is an inner layer called the endometrium and the muscular layer called the myometrium. I'll use those words are printed on another slide. what else? So then we can talk about the external female genitalia. This area here is called the vulva. So going from anterior to posterior, there's the clitoris that is not required for reproduction, but is part of the female reproductive system. It actually has a hood called the prepuce. So in the male reproductive system, the foreskin is called the prepuce, and in the female, the hood of the clitoris is called the prepuce. Same name for the different structures on the two male and female genitalia. The clitoris is drawn like this tiny little thing at the front of the vulva, but it actually is a large organ that has tissue that sort of goes around the on either side of the vulva. So it's not a tiny little thing. It's a sort of a Y-shaped structure. It's about that big. With my fingertips being the exposed part of the clitoris, and the rest of my fingers being the sort of embedded parts of the clitoris. And it actually has a corpus cavernosum, just like the male genitalia. It engorges with blood during sexual activity, and but there's not the same level of erection. The urethra is not part of the female reproductive system. It's part of the urinary system, so it's connected to the urinary bladder, but it's in the same vicinity. So I'm just pointing that out as a point of reference. And then the vagina is the opening from the outside to the cervix and the uterus. There are structures on the outside of the vulva, the labium minus or labium minora in plural, and the labium majus, labium majora plural. These are skin folds on the outside of the female vulva. So these are all anatomical terms to learn about. Questions? This is supposed to be the pubic bone, or the pubic symphysis, which is the fusion of two parts of the hip. Fat, anterior fat, posterior fat. There is a bulbourethral gland. There is also a bulbospongiosus muscle that undergoes contraction during female orgasm. Okay, so here is a schematic diagram of the most essential parts of female reproductive, rather than sexual characteristics. We're now talking specifically about reproductive organs. So here is the ovary, the female gonad, the fallopian tube, the fimbria that you can see in the previous slide is a more accurate drawing of the fimbria sort of wrapping around the ovary. Here the fimbria have been sort of pulled away from the ovary. The former is more accurate. There are ligaments that I'm not going to ask you to know the name of, but this one in particular is confusing because it looks like it's a connection between the ovary and the uterus, but it's just a connective ligament between the ovary and the uterus. And they have names, and frankly, I've forgotten. So I'm not going to ask you to learn them either. Inside the ovary is the developing ova, as well as a structure called the corpus luteum, which will be important in the female reproductive cycle. The uterus is this pear-shaped structure connected to the vagina. The cervix is the muscular area at the connection between the uterus and the vagina, which is closed. It is closed during pregnancy. It has openings, of course, when the woman is not pregnant, allowing sperm to enter from the vagina into the uterus and into the fallopian tube during fertilization. Which we're not going to talk about fertilization pregnancy, lactation. Today, we will do that after the next exam. It will be the one content class between exam 3 and the final exam. And it's sort of a summary of anatomy and physiology as well. So we're going to talk about the female reproductive cycle right up to the point of fertilization and pregnancy. So endometrium, myometrium. Myo is just, means muscle, smooth muscle. Oh, right. So we're not going to play the games, but there are, in the PowerPoint, you can cut and paste these links to the, into a browser and play the anatomy, ID game. And as before, let me just do it just to show you what we're talking about. Cut, copy, and paste. So, here's the midline sagittal section. Some of the names are names that I do want you to know, like uterus, and ovary, and labium minus, and clitoris, and, uterine tube is another name for fallopian tube. So you can use these games to practice your anatomy, but some of them are names that I do not expect you to know, like mons pubis. And rectum and anus are not part of the female reproductive system any more than they're part of the male reproductive system, so that they're not required. Helps you get oriented, but it's not required. So you can see some names that I do want you to know, and some names, some additional material that is not necessary to know. Let's see what the other one looks like. Looks the same, it's just got some different names. Prepuce, or the hood of the clitoris. Vaginal rugae. Now, those of you who've had A&P1 know that rugae are the folds inside the stomach. Well, apparently, there are folds inside the vagina also, which have the same name, vaginal rugae. Anyway. Some names that I do want you to know and some names that are additional, but it's an informative game to play. The names that I do want you to know are the ones from the coloring book, and, the coloring book also has a lot of structure that I don't expect you to memorize, so the words, your key words, the key concepts list, along with what's shown on slides like this, are the anatomical terms to know. Okay, so let's talk about oogenesis, the development of the ova. So there are parallels here, of course, to spermatogenesis, but there are some differences that are really interesting and important. So there are oogonia, primordial oocytes, and they develop in the fetus. So when the female is still a fetus, inside her mother's uterus, they develop into primary oocytes. And then so primary oocyte develops in, and they're in an arrest state. So they're arrested development from fetal development all the way through puberty. So the primary oocytes are in an arrest, meiotic arrest, until puberty. Then the hormonal changes that happen at puberty, age 11, 12, 13, start the process of, or continue the process of oogenesis. So oogenesis starts in the fetus, is arrested for 12 years or so, and then is allowed to continue during puberty. After puberty, meiosis I happens, So this is going from a diploid stem cell or precursor cell into starting to become a haploid gamete. But what's interesting is that in the male, 2 developing cells arise from this meiosis 1. But in the female, there is 1 secondary oocyte and 1 sort of residual cell called a polar body. So 2 cells develop. They are haploid, but only one of them is the secondary oocyte. The other one is sort of a sideline. And this secondary oocyte is an oocyte that can develop into a mature ovum if it is ovulated. And if it becomes fertilized, it undergoes meiosis 2. And again, in meiosis 2, there is 1 mature oocyte that has actually been fertilized now. And it generates another polar body. The first polar body, which is really a vestigial thing anyway, also undergoes meiosis 2 to develop 2 more polar bodies. The bottom line is that in oogenesis, 1 primary oocyte develops 1 haploid before fertilization, mature oocyte, even though it goes through the 2 stages of meiosis, just like in spermatogenesis. So in spermatogenesis, we generate 4 haploid gametes, but in human oogenesis, we develop only 1 gamete, 1 ova, even though it's gone through 2 stages of meiosis, because there's also the generation of 3 polar bodies, which are not ova. And the other interesting thing is just how long this is delayed, right? We have a 12-year delay here, and we have meiosis 2 happening only after the oocyte becomes fertilized. And then, of course, after fertilization, development of the embryo occurs. So there's the second meiosis 2, and then combination of the chromosomes from the sperm and the egg, and development of the embryo. So now let's transition to talking about the human reproductive cycle. This is not true of all mammals. This is true of human female reproductive physiology and anatomy. So I guess we could start at number 1. Day 1 is when menstrual bleeding starts. That's just considered the start of the female reproductive cycle. So the beginning of the menstrual cycle or the beginning of uterine bleeding is day 1, and this is from the beginning of the menstrual cycle to the point of ovulation, to the moment of ovulation of an oocyte, is called the follicular phase. Okay, so the first half of the 28-day female reproductive cycle is the follicular phase. And during the follicular phase, actually, as the uterus is shedding its endometrium, uterine bleeding, the the ovary, is allowing multiple follicles, each follicle containing an oocyte, to develop. Multiple follicles develop during the beginning of the follicular phase. And I'll show you another slide that has this more in a graphic cartoon version. This is the timeline version. So we got a timeline version, and then we'll see a graphic of the, I'll just show you what's coming up. A graphic of the ovary coming up. So let's go back to the timeline. Uterine bleeding, during which multiple follicles develop. One follicle is going to become dominant, and the other follicles that have started to develop undergo atresia, or apoptosis, and don't continue to develop. One follicle sort of takes the lead, and the rest don't. One follicle becomes dominant, and it matures, and the rest undergo atresia. At 14 days in the cycle, that follicle is released from the ovary, and that's the point of ovulation. The ova, the egg, travels from the ovary, picked up by the fimbria, transported by sort of wave-like ciliary movement in the fallopian tube from the ovary to the uterus. If there's going to be fertilization, it's usually here during fallopian tube transport. At the moment of ovulation, the female reproductive cycle transitions from the follicular phase to what's called the luteal phase. And what you'll see is that part of the follicle that is not ovulated becomes the corpus luteum. And the corpus luteum is an essential part of the endocrinology of the female reproductive system, especially if there's a pregnancy. And if there is not a pregnancy, the corpus luteum does not get the signals to sustain itself. It undergoes apoptosis and degeneration. And then the degeneration of the corpus luteum is what leads to the release of the highly developed uterine endometrium, and menstrual cycle begins. Remember, this is drawn as a linear timeline, but it's a cycle. So we connect from the end here, the 28th day, right back to the first day here. Katie?
It's just the follicle phase, is it just like when one becomes down, such as because like
we have two ovaries.
Yeah.
So so it's an interesting thing. The ovaries typically, not always, but typically the ovaries sort of go back and forth. And one of them is in sort of a less, so the dominant follicle is the dominant follicle in one ovary. And the signals that cause one ovary to say, okay, it's my turn, and the other ovary to say, yeah, it's your turn, and then to switch, I don't know what those signals are.
Just like with fraternal births, like then.
Fraternal twins, you mean?
Yeah.
Yeah, so if both ovaries do it, and they both release an oocyte, and both of them become fertilized, that would be dizygotic or fraternal twins. Yes?
Will you explain the importance of the corpus luteum?
I will. Hold that thought. So here's a cartoon from your textbook that shows the changes in hormones. These are hormones, LH and FSH. These are hormones, estrogen and progesterone, during the menstrual cycle. So going from the follicular phase to the luteal phase and then back to the follicular phase. So remember, all of this is cyclical. So here, these complicated changes in hormone levels are something I will make sense of. But let's start down here with the physical developments of what happens in the ovarian follicle, in the ovary. It's all happening at the same time as these endocrine changes. So what I want to do is start with something rather simple, the physical changes, and then get to something more complicated, the endocrine changes, but they're all happening together. And maybe I'll cycle around and say how these endocrine changes then cause the physical changes that I'm starting with, okay? So I'm going to start here, then go here, and then try to tie it off. So I'm being cyclical as well in terms of my teaching. So let's talk about the physical changes that go on in the ovary during the follicular phase. This is during the follicular phase. And again, just like the cartoon that we had of the seminiferous tubule, where the sperm seemed to be moving laterally as time was happening. That's not what's happening here. So, for example, this looks like these primary follicles are moving from here to here as they become secondary follicles and then mature follicles and then undergo, what's it called, ovulation. But that's not what's happening, right? So we're trying to compress time into one physical space. And so if this oocyte is the one that is going to become the secondary follicle that dominates and becomes the mature follicle, this follicle and this follicle are happening at the same location. There's no movement of the follicle around the ovary. This is just trying to compress a bunch of events in time in one cartoon. Is that clear? So the point is, let's just start over here. So we're going around this cycle, starting here, but there's no physical movement. This is just movement in time, not in space. Is that clear? So at the very beginning of the menstrual cycle, there are primordial follicles. In each of those follicles is the oocyte. So the follicle is a structure that contains the oocyte, but is not just the oocyte. I'll talk about some of the cells as well. So there is the primordial follicle that developed during the fetal stages and then went into meiotic arrest. Let's skip this one here. This is actually the end of the cycle, so we're going to go from here to here. The primary follicles, some of them develop during the follicular phase, triggered by changes primarily in estrogen. Bringing in the endocrinology, but we'll focus on endocrinology in a few minutes. So some of the follicles that are primordial become primary follicles. You can see they get bigger, the oocyte gets bigger, and the cells surrounding the oocyte gets bigger. The cells, so now let's talk about the cell types of the follicle. So inside the follicle is the oocyte. Surrounding the oocyte is a layer of cells called granulosa cells. This is an important name to know, granulosa. And on the very outside of the oocyte, even though it's not labeled till down here, these cells on the outside of the follicle, outside the layer of granulosa, outside the layer of the granulosa is the theca, or thecal cells. And on this slide, they're called theca follicula. So we have oocyte, granulosa, theca. There's going to be connections between the granulosa and the Sertoli cells and the theca and the Leydig cells. Just saying, so stay tuned for that. So primordial oocytes develop, the oocyte develops, that is to say it gets larger in response to endocrine signals. The granulosa cells proliferate in response to endocrine signals. The theca proliferates in response to endocrine signals coming. There is a space inside the follicle called the antrum that is filled with a rich fluid of hormones and other things. So here is the mature follicle, a mature oocyte surrounded by granulosa cells, a antrum, more granulosa cells, and a theca, a layer of thecal cells. At the moment of ovulation, there is release. This looks like exocytosis, but this is not exocytosis because this is a cell being released from an organ. This is ovulation. Ovulation is the release of the oocyte surrounded by some of the granulosa cells, which are now called the corona radiata, and the rest of the granulosa cells and theca cells become the corpus luteum. The corpus luteum continues to develop in response to endocrine changes and will maintain itself as an important endocrine organ if there is fertilization. And if there is not fertilization, then the signals for sustaining it don't happen, and it degenerates. And that degeneration then is accompanied with uterine shedding and bleeding. Michael?
Does this all happen
Pardon?
This is all happening in the ovary.
This is all happening in the ovary. I mean, the endocrine changes are happening elsewhere. Some of the endocrine changes are happening elsewhere, but what we're talking about here is in the ovary. So let's see if there are any other anatomical terms that I do want you to know. Here is the name of a ligament that I don't care that you know. We've got a name that you already learned from male reproductive anatomy called the tunica albuginea, the layer on the outside of the ovary, which is the same name as the layer on the outside of the testes. I do want you to know the name primordial follicle, primary follicle, secondary follicle, granulosa cell, and oocyte. Antrum. Okay, here's another one. There is a layer between the oocyte and the granulosa called the zona pellucida. This layer between the oocyte, in this case the ovulated oocyte, and the corona radiata, which are granulosa cells, this layer is the zona pellucida. Theca, corpus luteum, pretty much summarizes it. The ovary has a cortex and a medulla that I don't need you to commit to memory, but just so you know, it's true. Yeah?
Causes that, but is it primarily because of like when you're a fetus and the bowel issue, or is there an issue with this process, or can you like, you have some work or
Yeah.
things?
So, fertility problems, is that what you're asking about?
Yeah.
So, it's, I'll bet there are problems at each of these stages. Development of the oocyte, release of the oocyte, transport of the oocyte, additional barriers to fertilization that happen, that prevent sperm from getting to where they need to get to, barriers to fertilization of the ovulated oocyte, genetic problems that cause, miscarriage before a person, not miscarriage but an oocyte might get fertilized, but never gets implanted, and in fact doesn't, there is in fact a fertilized oocyte, but if implantation doesn't happen, then a menstrual cycle will happen, and you would never know that there was a fertilized oocyte. So there's going to be complicated reasons for fertility issues. Yes, Zoe.
So, how does the primordial follicles turn into the
So the primordial follicles are they're sort of under control because there's low levels of estrogen, but during puberty we're going to start to see estrogen changes. Estrogen, that I'm going to talk about real soon, are going to be triggering some of these cells to undergo, let me go to the next slide. So the primordial follicles are responding to and producing estrogen. So we're going to see a feedback thing going on here. Estrogen is going to be both a response and a cause. Well, a response and the follicle are going to be producing estrogen and responding to estrogen. And some of the follicles' response to estrogen will be to continue to develop, to enlarge and to proliferate the granulosa. So the short answer to your question is estrogen. Why some and not all, and why one becomes dominant and others then undergo atresia is complicated, and in fact I can't tell you. I don't know the molecular mechanisms for those things. Alex?
With the corpus, how do we were thinking about like when you get pregnant, it knows because of what?
Yeah. When you get pregnant, it knows that you are pregnant. And that causes the corpus luteum to be sustained, to continue to grow, to suppress any more oogenesis, and to lead to the changes that are associated with pregnancy. We'll talk about, at the end of this class, we will talk about how the corpus luteum knows you're pregnant. There's a hormone that's released from the uterine endometrium that tells the corpus luteum, I'm pregnant. Yeah.
Is that what's shown, like, if you don't become pregnant, is that?
If you don't become pregnant, then that hormone from the uterine endometrium doesn't happen. The signal for the corpus luteum to maintain itself doesn't happen, and the corpus luteum degenerates. Hormone levels change as a result, in particular progesterone and estrogen levels change as a result of the degeneration of the corpus luteum, and that's what causes the uterine lining to lose its stability and to become shedded in menstrual flow. I'm glad for these questions, even though we're getting out of the linear sequence, because it is all a cycle. And so seeing what's coming up can be helpful to understanding what's going on now. So this is just a cartoon of oogenesis. It outlines some of the names of the structures that I do want you to know. The primary follicle with the developed oocyte, with the zona pellucida separating the oocyte from the granulosa cells, the oocyte, zona pellucida, granulosa cells, and theca cells developing, the oocyte, zona pellucida, granulosa, a fluid-filled antrum, and a layer of theca, and finally the mature follicle, ready to, this is the dominant mature follicle, ready to be ovulated, or ready to undergo ovulation. Going from the outside this time, theca, granulosa, antrum, zona pellucida, oocyte. Here's a name of a structure, the cumulus oophorus. It's not on your list. So this is the life cycle of the one follicle that became dominant. The other follicles that don't make it probably degenerate somewhere in this part of the cycle. They certainly don't get to the antral stage of development. And they are, so they degenerate, they are absorbed by the cortex of the ovary. Yeah. I don't know the molecular mechanisms for dominance. I'm not sure we, as a society of knowledge, I'm not sure that it's well known. But I could look that up. I know I don't know. Okay, I think that, well. So here are some images of the way things really look in stained microscopic section. So here is the edge of the ovary. Here is the tunica albuginea. Here are primordial follicles. So this is a high magnification looking at primordial follicles. Here is a primary, not primordial, a primary follicle with the oocyte, zona pellucida, and granulosa layer. Here's a primary oocyte, which is just really primordial follicle that has the oocyte in it. So, words to know: zona pellucida, oocyte, primordial, primary, follicle, and granulosa. Continuing, the primary dominant follicle then becomes the antral follicle, or the mature follicle, that has the oocyte, the zona pellucida, granulosa cells, a fluid-filled, not a cellular, a fluid-filled antrum, and the theca, the layer of thecal cells outside the granulosa cell layer. Zooming in here, oocyte, zona pellucida, granulosa, theca. So the cartoons are representing, things that actually look like this. And here is an image of ovulation. Bursting of the tunica albuginea, releasing of a follicle, releasing of an oocyte, surrounded by the corona radiata, which is really a name of the layer of cells that surrounds the oocyte, granulosa cells. But now, once it's released, it's got a special name called the corona radiata that I do want you to know. Here's the external surface of the ovary. Here's a word that's not on your list. Here are the fimbria, the fingery structures at the beginning of the fallopian tube, or the uterine tube, also known as the uterine tube or fallopian tube. They're synonyms. And typically this space is small. I think to get this image, they probably expanded this space with injection of fluid or something. But typically the distance between the fimbria and the ovulated oocyte is very small, and the fimbria then bind and transport the ovulated oocyte. There are more games to play. Let's just see what these look like. We're not playing the games because I'm concerned about time. I just want to make sure that we get to everything that we need to get to. But the games can be fun. Right, so it's images of the ovary and the development of a primary follicle. So again, using the language in a game that makes a pleasing sound. Corpus luteum. Have fun. Okay, let's take a short break here and start to talk about the hormonal changes during the female reproductive cycle, during the menstrual cycle. We'll come back and connect the endocrine changes to those physical developmental changes. Talk about endocrinology. So this is a cartoon of the endocrine changes during the follicular phase and the luteal phase of the female reproductive cycle. So this is, I trimmed so I could enlarge this one, but this is follicular phase from 0 to 14, and this is the luteal phase from 14 to 28. And we'll label your handout to make that clear. Together, it's the menstrual cycle, follicular phase, luteal phase. So during the follicular phase, there is low but increasing levels of LH and FSH. We'll talk about that. The trigger for ovulation is something called the LH surge, which is accompanied by a smaller surge in FSH. So luteinizing hormone and follicle stimulating hormone have this spike of level of concentration that is a signal of, but really a cause of, ovulation. There are also changes in estrogen and progesterone that are important during this cycle. So let's start during the, say, sometime during the follicular phase. Just during the follicular phase. Remember, a follicle is developing, one is becoming dominant, the oocyte is getting bigger, the granulosa cells are proliferating, as are the theca. That's what's happening during the follicular phase. During this follicular phase, so this is the early and middle follicular phase, the hypothalamus is secreting GnRH in response to cues. GnRH is released into the median eminence, into the portal circulation, and it triggers the gonadotrophs of the anterior pituitary to secrete FSH and LH. Now, things are going to look a little bit like what happens in the testis. FSH stimulates the granulosa cells to develop, similar to the way FSH stimulates the Sertoli cells to develop and promote spermatogenesis. LH causes the theca cells to develop and to release androgens, similar to how LH triggers the Leydig cells to release androgens like testosterone in the male. So there are some similarities here. So FSH is stimulating the proliferation of the granulosa, and LH is triggering the developing theca to release androgens. Androgens are released locally, so they are both endocrine and paracrine molecules, and when acting as local, when the androgens diffuse to the granulosa cells, the granulosa cells, which are being developed in response to FSH, convert the androgens into estrogens. Theca releases androgen, just like Leydig cells release testosterone. Theca, the androgens from theca cells are converted by the local granulosa cells into estrogen. And estrogen then circulates throughout the body. Estrogen is going to contribute to the development of all of these cells, as well as to other, systemic changes in estrogen. Okay, so that is the feed-forward story of endocrine changes in the follicular phase. There is feedback inhibition. Estrogen gives negative feedback to help keep LH and FSH levels under control. Estrogen has negative feedback to GnRH to help keep GnRH and LH and FSH levels under control. Low, but under, not zero, but under control. Negative feedback. There is an inhibin also that, let's just leave that aside here, just like we left it aside in the male reproductive system. There's another hormone that is important for negative feedback. The point is there is negative feedback during the follicular phase, keeping LH and FSH levels not zero, but at a set point. As the estrogen levels get high, so why are estrogen levels getting high? The theca are releasing androgens, but remember the theca are also proliferating. So they're making more androgens. The androgens are being converted by the granulosa into estrogen, but there is more granulosa. The granulosa cells are proliferating. So the proliferation of these cells leads to more and more estrogen. Small, increasing levels of estrogen. Until we get to a tipping point. There is a tipping point at a concentration of estrogen at which when the estrogen levels are high, because the follicle is now large, fully developed, produced the theca of the follicle is producing a lot of androgen. That lot of androgen is being converted by the granulosa into a lot of estrogen. When estrogen levels get high, the negative feed, and this is always the strangest part, the negative feedback that was happening previously by low levels of estrogen now get converted into positive feedback. There are low affinity receptors in the hypothalamus and in the gonadotropes of the anterior pituitary. And when those low affinity receptors are activated by high amounts of estrogen, high concentrations of estrogen, a new process starts, and that's positive feedback. So now estrogen, instead of suppressing LH and FSH release, is now positive feedbacking to stimulate LH and FSH release, and that's the LH and FSH surge. And that large amounts of estrogen combined with a spike of LH and FSH is what triggers ovulation. So I'm trying to combine the physical and the endocrine changes here. So we have explained how late in the follicular phase, as estrogen levels get high because of the developing follicle, developing theca, developing granulosa, higher and higher amounts of estrogen, at a certain tipping point, the negative feedback, keeping LH and FSH low, is flipped into positive feedback, and we get an LH and FSH surge. And that LH and FSH surge, combined with high levels of estrogen that cause the LH and FSH surge, is also what causes ovulation. Yeah.
What causes the change between the negative feedback and the positive feedback?
I believe it is complicated, but I think one of the simplest things that must accompany it is that when we have small amounts of estrogen by the less than fully developed follicle during the follicular phase, low amounts of estrogen are interacting with high affinity receptors, triggering negative feedback. But development, more and more cells, more and more release of estrogen, high amounts of estrogen are interacting with low affinity receptors in the hypothalamus and the anterior pituitary. And those high affinity receptors, those low affinity receptors are different. They trigger different genetic changes in these cells, different transcription factors, and they then say, okay, make more LH and FSH. Okay, so now what terminates this surge is progesterone. The corpus luteum, the structure of mostly granulosa cells left over after ovulation of the oocyte and the corona radiata, the corpus luteum produces large amounts of estrogen, as it did before, but also with progesterone. The combination of progesterone, this is now a different hormone in the mix. Now we've got a new hormone in the mixture of hormones, the progesterone and estrogen. Even though this is high estrogen, we now have high progesterone, and that somehow the progesterone overrides the positive feedback of high estrogen and converts back to negative feedback, which is why we get a termination of the LH and FSH surge and a return to negative feedback, because we have high levels of estrogen, but we now have really high levels of progesterone, and the combination of estrogen and progesterone suppresses the LH and FSH surge because of a return to negative feedback. So the corpus luteum, the leftover after the ovulation event, is what returns the system back to negative feedback and puts LH and FSH levels into, pre-ovulation levels. Progesterone is one of the keys. and let's just look at, it looks like estrogen and progesterone are at about the same levels. Well, let's look at the scale here. Progesterone, nanograms per mil. Estrogen, picograms per mil. So the progesterone scale is 1,000 times higher than the estrogen scale. So progesterone levels are 1,000 times, they're not equivalent, they're 1,000 times higher. We're talking about 15 nanograms per mil versus 15 picograms per mil. Remember, there are 1,000 picograms in a nanogram. Yeah.
The progesterone, is that going up right when you have ovulation? Because I mean, like, we have the decrease,
Yeah.
the sharp decrease then.
So at ovulation, there is now there is a corpus luteum. And now this corpus luteum is starting and continuing to make large amounts of progesterone. And this large amount of progesterone is what triggers the return to negative feedback. Okay, so going forward, this is where we were. Corpus luteum, huge compared to estrogen amounts of progesterone. Return to negative feedback. So the corpus luteum is the thing during the luteal phase. There's the name of it. The corpus luteum is the most important thing going on during the luteal phase. Development of the follicle is the most important thing during the follicular phase. And the names give you a little bit of information about what is happening during those stages. The corpus luteum then, and this enormous amount of progesterone that it's producing, enormous compared to estrogen, nanograms per mil is still a pretty low concentration, so everything's relative. But the large amounts of progesterone relative to the estrogen suppresses the LH and FSH surge and allows the continuing development. It sustains both the corpus luteum and prevents another ovulation event by preventing another LH and FSH surge. The birth control pills, female birth control pills, some of them are complicated mixtures, but they all contain large amounts of progesterone. So here is a summary. During the follicular phase, this is a summary really focusing on the endocrinology, but remember the endocrinology and the physical changes are all linked. During the follicular phase, these little star things are supposed to be hypothalamic neurons that are releasing GnRH into the median eminence, causing the anterior pituitary gonadotropes to release LH and FSH, which stimulate. This cartoon there's an oocyte. Here is the layer of granulosa. This is supposed to be the developing theca. The theca releases androgens. The androgens are converted by the granulosa into estrogens, and the estrogens are triggering changes, development of the follicle, and negative feedback. Just before ovulation, there are high levels of estrogen because the cells have proliferated into this complicated structure, large amounts of fecal cells, large granulosa converting the androgens into estrogens, high concentrations of estrogen triggering a positive feedback event, leading to a spike in LH and FSH, which causes the mature follicle to be ovulated. After the ovulation, the corpus luteum, which is different than the mature follicle because there's no more oocyte inside it, and so it knows it's different. It now starts to release large amounts of progesterone, and the large amount of progesterone converts the positive feedback back to negative feedback and suppresses LH and FSH and prevents ovulation. The female, the birth control pill The high amounts of progesterone are preventing ovulation. Questions? So let's just talk a little bit more about the estrogens and the progesterone as hormones. You know, just some sort of tie it back to endocrinology. Estrogens and progesterone are both steroids, so you can go back to that complicated flow chart and you can see that estrogens are, well, they're all derivatives of cholesterol. They all are downstream of progesterone. The estrogens are all downstream of the androgens. They're steroids. The stimulus for the stimulus for estrogen release, and they are released by the cell, the ovarian follicle, by the granulosa of the ovarian follicle, and by the corpus luteum. The cells that release it are these, and the triggers for the release are LH and FSH. LH and FSH cause the development, participate in triggering the development of the granulosa and theca, as well as the release of estrogens and progesterones. There are many target cells. The receptor is the receptor, both the low affinity and the high affinity receptor for estrogen are intracellular. And so what are the effects of estrogen? Well, I mean, female body characteristics, breasts, hips, lack of body hair, convert to male, female, you know, what am I trying to illustrate here? Maternal behavior. So what am I trying to illustrate here is body type. What am I trying to illustrate here? Maternal behavior. What am I trying to illustrate here? I don't know. It's a painting by a very famous Dutch painter, Vermeer. I thought it was beautiful. I am not suggesting that it is typical for women to look like this, or is required for women to do this. I am not suggesting that women need to do this. That just to be clear here, I mean, I think I included some other slides. That, you know, women can do this, and women can discover black holes, and women can be astronauts, etc. Right? But you know, there are differences between men and women, and those are reflected both genetically and hormonally. So the other hormone to talk about is progesterone. It is also a steroid. It is released by the corpus luteum. Its trigger is also LH and FSH, but now you need the corpus luteum to release the large amounts of progesterone. Its target cells, so now during the luteal phase, the progesterone is maintaining the uterine endometrium. The progesterone is causing the mammary gland to develop, not necessarily to produce milk yet, because we are not, there is not a fertilized oocyte, there is not a pregnancy yet. I'm going to talk about that after next week. But the mammary gland develops in response to both estrogen and progesterone, especially these big pulses. Remember, every single menstrual cycle is accompanied by this big pulse of progesterone. And so with each of those, there is a trigger for the development of the female mammary gland. So progesterone's effects are to maintain the uterine endometrium and to grow mammary tissue. So here is a final slide that sort of summarizes, let's take this one apart. This is a summary slide. We're in the follicular phase. A follicle develops and becomes dominant. So I can break down these steps, but remember, a follicle with granulosa and a zona pellucida and some primitive fecal cells is developing. LH and FSH are under negative control of the low levels of estrogen. And this part here, which is new, this is supposed to indicate the thickness and development of the uterine endometrium. So here the uterine endometrium is degenerating during menstrual flow. Release of the cells of the uterine endometrium. So, and then after menstrual flow is finished, the uterine endometrium starts to develop again under the stimulus of increasing amounts of estrogen and small but non-zero levels of progesterone. Uterine endometrium developing, that is to say thickening, getting more and more vascularized, preparing itself to become implanted with a fertilized oocyte, getting ready for pregnancy. Developing endometrium is happening at the same time as the developing follicle. At a certain point of estrogen concentration, estrogen levels are increasing because of the proliferation of cells. Estrogen levels increase to a point where they no longer do negative feedback on LH and FSH, they do positive feedback and cause the spike. The spike causes ovulation. The ovulation then causes the creation of the corpus luteum. The corpus luteum is now making enormous amounts of progesterone that continue and to allow the uterine endometrium to develop and to sustain itself. Large amounts, this is progesterone, but again, the scale is wrong by a factor of 1,000. Large amounts of progesterone, 1,000 times higher than this relative to estrogen. High levels of progesterone, high levels of estrogen, maintenance of the uterine endometrium. That was caused by the corpus luteum. The oocyte, the ovulated oocyte, travels in the fallopian tube. It doesn't travel, it is carried by the fallopian tube. Movement of the cells, the cilia of the fallopian tube, move the oocyte from the fimbria at the beginning to the uterus. If there is no fertilization, so that's what's going on here. If there is no fertilization, the corpus luteum does not receive a signal to sustain itself, and so it undergoes degeneration. Progesterone levels plummet. The uterine endometrium loses the hormones necessary to sustain it, and it loses its stability, and it sheds the uterine lining, the uterine endometrium. Not all of it, but there is shedding of the proliferated cells of the uterine endometrium, and that's menstrual flow. Okay, so we've done a non-pregnancy cycle. This now is a pregnancy cycle, and we're not going to talk about it until next week. The key difference is that if there is implantation of the fertilized, so here's fertilization, if there is implantation of the fertilized blastocyst, primitive embryo, if there is fertilization, the uterine endometrium releases a hormone called chorionic gonadotropin. And for humans, it's called HCG, human chorionic gonadotropin. This is a hormone that tells the corpus luteum not to degenerate. And it maintains the corpus luteum. Levels of estrogen and progesterone are high. Remembering that progesterone levels are 1,000 times higher than this relative to the level of estrogen. The corpus luteum sustains for the next 9 months of pregnancy, maintaining the uterine lining, suppressing any further ovulations, causing further development of the mammary gland, getting ready for childbirth. Other physical changes, softening of bone, widening of hip, etc. The trigger is this, human chorionic gonadotropin. The pregnancy tests that the kits that you can get in pharmacy to measure, are you pregnant or are you not pregnant? They are basically a Western blot for chorionic gonadotropin. If this hormone is released, it's the sign of pregnancy. If it is not released, it's the sign of not pregnancy, and pregnancy doesn't happen. So this is the part to understand for exam 3, and we will get to changes related to this after exam 3. Questions? I had time, I could have played the game. Yes?
So how does that hormone, like, any pregnancy test go, like,
So it's a hormone. It's actually a peptide hormone. It's a small protein. It gets into the blood. Apparently enough of it gets into the urine. And the antibodies we can use for those little stick tests are good enough to be able to detect the femtograms or picograms per mil of that hormone in the urine. It gets into the urine, by the way, that everything gets in the urine through the kidneys. So that's not its target, but there's enough, it gets into the bloodstream and it gets into the urine from there. Yeah.
Also, I don't know, but like, how is this changed if a woman has
Endometriitis, yeah, that, I haven't looked at it. I don't know, so, yeah, I don't know the answer. So there are some clickers here that you can play. Why don't we just say you can use these at home to review. The answers are on Moodle. I'll make sure this PowerPoint is available to you on Moodle right now. So the homework, if you for studying for this, are PowerPoint, reading it from the textbook, clickers, and the happy little Bing game. I'll see some of you in lab this afternoon.